Hello dear students, welcome again to another lecture. In this lecture, I will be discussing a very important topic, that is the Lac Operon or the lactose operon. Now, firstly we must know meaning of two terms. That first is the lactose. Okay, so what is a lactose? Lactose is nothing but a disaccharide. Okay, when two monosaccharides are binded, they form a disaccharide. Okay, similarly glucose. What is glucose? Glucose is a monosaccharide. Okay, glucose gives us energy. Now lactose also gives us energy. We know that lactose can be found in milk. Okay, now what happens is that when the bacteria are not getting this glucose, okay, they're not getting the glucose, what will happen? They will starve to death, no? They will die. But not always. If they are having a alternate source, that is the lactose, what the bacteria will go to this lactose and obviously survive on it. Clear? So this Lac Operon is a very much important topic, and obviously it is a bit of difficult, so I will be teaching you very slowly. Now what do you mean by operon? Okay, so let's discuss the meaning of operon. Now operon is a cluster of similar genes. That means that similar genes are present. Okay, we all know that in a DNA, suppose this is a DNA, a fragment of DNA contains gene. Gene produces protein, right? We all know all this. Okay, so the next thing is, inside the gene or inside the DNA, what we found? We find some components. Okay, so now we must know about the components of Lac Operon. Okay, what are the components of Lac Operon? What this Lac Operon is made up of? It is made up of a regulator gene. Okay, remember, this regulator gene produces repressor protein. Okay, just I am writing that repressor protein. Clear? Now next comes what? Next comes the promoter. Okay, we all know inside the DNA segment there is a promoter. Okay, where the RNA polymerase binds. Okay, so there is a promoter. Now before watching this lecture, you must go through the transcription replication process. Okay, or else you will be not able to understand a single word. ठीक है ना? Now RNA polymerase binds in the promoter sequence. Number 3, see, there is a operator. Okay, this operator comes just after the promoter sequence. It is just a switch. Okay, it is a on-off switch. Now just see the concept. When glucose is available in the system, suppose this is a Petri plate. Okay, just let me delete all this. Suppose this is a Petri dish or a Petri plate. Okay, this is a Petri plate. or petri dish. In the petri dish, you have kept glucose. Okay. Now, what will happen? Suppose the red things are glucose, okay, and you have cultured bacteria. You have cultured E. coli bacteria. These are E. coli bacteria. Okay. So, this E. coli bacteria will grow, divide in number when there is this nutrition source. Okay. Right now. But when there is no glucose, suppose, and you have also kept here another source of nutrition, that is the lactose, okay, which is present in milk. What will it do? Suppose glucose is absent in this petri dish. Lactose is, I am denoting lactose by this violet one. Suppose lactose is also present in this medium, but glucose is not present. This bacteria has the gene clusters. And operons are what? Operons are cluster of similar gene. Remember, it is very important. Okay. So this E. coli will turn on what? Its Lac Operon genes, and it will be able to intake or break down or metabolize the lactose. Okay. So just think of it. If in the system, if in the petri dish there was glucose, okay, इसमें suppose glucose present था, okay, then what happens? Lactose operon का कोई ज़रूरत नहीं है. There is no need of running this Lac Operon that I am teaching you. Okay. So क्या होगा? Lactose operon कब ऑन करना पड़ेगा? When the E. coli or the bacteria will turn it on when there is no glucose in the system. Clear? Now, I think you have understood. Now, there are the structural genes, that are the main genes which are required. Okay. So, that is the Z gene that produces the beta galactosidase, which breaks lactose. Okay. Lactose is a disaccharide, it needs to be broken, right? Then comes the Y. The Y gene, Z is a gene, Y is also a gene. Okay. It produces permease. Okay. That helps lactose to enter the cell. Obviously, this lactose which is outside, it must enter the bacterial cell, right? It must what? Enter the bacterial cell. Okay. It must enter the bacterial cell. This Y gene produces permease. Clear? That helps it to enter the bacterial cell. And then comes the last, that one is the A gene. It produces, it is not that much important, and the research is still going on. What is the function of A gene? So A gene produces trans-acetylase. Okay. So there are 3 genes. These are the structural genes. I am mentioning it. These are the structural genes. Okay. So how to remember the genes? These are ZYA, ZYA. Function of Z gene is breaking the lactose. Okay, you must not break it, na? You need something to break it because it is a disaccharide. It will be broken. Now lactose can be broken into remember, glucose. It can be broken into glucose and galactose. Okay. I will be showing a diagram also to you all. Clear? So what happens at what time? See. Now this inducer gene which I was talking about, it produces what? It produces a repressor mRNA. Now see, when there is what? There is glucose present in the system, what will happen? The E. coli is very smart, not like us. Okay, is very smart. जब सिस्टम में या पेट्री डिश में ग्लूकोज प्रेजेंट होगा, okay, this I will be, inducer gene will be turned on and repressor mRNA will be produced. Okay, means repressor protein, this is a protein named repressor protein. repressor protein कहाँ से प्रोडक्शन हो रहा है? From this I gene. Okay, inducer gene is producing a repressor protein. Now this repressor protein, what will it do? It will just bind to the operator. Okay, and see, repressor bind to the operator region and prevents RNA polymerase from transcribing. That means that RNA polymerase, suppose this is the RNA polymerase will not move further. And if what happens if RNA polymerase does not move further, there will be no production of this ZYA genes. Okay, now this happens when glucose is present. That is why I am writing plus. Okay, now see, in the absence of glucose and obviously in the presence of lactose, see, the repressor gene becomes inactive. Why? There is another thing to learn. Because there is an inducer present named as allolactose. Okay, this inducer, remember, this inducer binds with the repressor, okay, and makes it inactive. मतलब एक inducer present होगा, वो भी lactose से ही आता है. Okay, that inducer's name is allolactose. This inducer binds where? This inducer binds to the repressor mRNA. Obviously, repressor mRNA कहाँ bind हो रहा था? Repressor mRNA was binding to operator region, and what? It was blocking the RNA polymerase to go forward and transcribe. तो operator में यहाँ पे repressor नहीं लग पाएगा, and smoothly RNA polymerase will be able to what? It will be able to do the transcription and produce the beta-galactosidase protein, permease protein, and trans-acetylase protein. Okay, so this was the thing. Okay, so it is a very easy thing. Means I have described it very easily. I feel that. Okay, so that was it. Only in 15 minutes you have learned the whole Lac Operon concept. And remember that Lac Operon was proposed by, because NEET asks this type of questions. Lac Operon was proposed by Jacob and Monod. Okay, remember it. And it is found in prokaryotes only. It is not a eukaryotic gene regulation. Clear? So that was the fact. And allolactose acts as the inducer. I have told it. Clear? So I have one question for you all. Can you answer it? Okay, I am writing it. Please try to answer. Lac Operon is A, repressible, B, constitutive, C, inducible. Okay. Write the correct answer in the comment section. Okay, thank you for watching.